This is the final prep for lecture 29. And so this is just a point about the whole process of limb development. So um, the patterning signals for both limbs are similar, but each one, the materials from each of the different limbs, forelimb, hindlimb, interpret those signals uh, to produce different limbs, especially noticeable in the chick, the wing versus the leg, right? Very different kinds of limbs, but the proximal, distal, anterior, posterior, and all that, those kinds of signals, those are the same. But the identity of the limb is specific. It doesn't change. So I talked before about grafting a bit of the proximal mesoderm from the leg bud in a chick to a distal region in the wing bud. And so when that tissue was took part in the development of the limb, it still formed leg structures, but it formed distal leg structures. So it kept its limb specification. So it was determined to form the leg bud, but it was only specified to form proximal because when you moved it to a more distal region, it was able to change. So this can be potentially confusing, but what it means is that you can separate sort of the identity of each of the limb buds from the patterning signals that are the same in both. Okay, and it's the TBX factors, remember TBX4 and 5 and PIDX1 and so on, that tell, the limbs, okay, you're going to be forelimb, you're going to be hind limb, but also the Hox genes are involved, and we haven't really talked very much about them, okay? But so basically, this is just what I've been saying. And so the only thing you really need to know, I'm not going to go into the Hox genes, but they play roles in both anterior, posterior, and proximal distal specifications. So they are responsible for determining sort of the forelimb versus the hind limb, but also as the limb is growing out and the parts are being specified, there is Hox gene expression that differs from proximal to distal that helps to specify those sections. This part of the text we're leaving out, and so this is the only part you need to remember about Hox genes. I want to finish by talking about programmed cell death. It's in order for you not to have webbed hands and webbed feet, there needs to be separation of the digits from one another. And that means that the cells between the digits die. If you look at your own fingers, you can see just a little bit of webbing down at the very base where the fingers meet each other. And some people have webbing that extends a little further and others that extends less far. And that's just a measure of how much cell death took place while your hands were developing. Okay. And just a point I've made a couple of times in class is that programmed cell death is a feature of development. It seems weird to have dying cells as a whole program of development, but that does happen in more than one type of tissue, notably the neurons. When embryos are developing, they produce way more neuronal cells than are needed, and the ones that don't connect up and become part of the whole peripheral nervous system and central nervous system they're culled, they're removed through programmed cell death. And so BMP signaling in the interdigital space is required for the apoptosis, and this is a separate, it works after it's contributed to digit identity, so this is two separate things. And lack of BMP signaling in the interdigital space in the chick embryo at least leads to chicks with webbed feet. Which doesn't hurt them. They can survive. It's not a lethal thing. And it's the mesoderm in this space that is where the apoptosis gets induced. So this is the 1, 2, 3, 4. So this is a mouse limb developing, and you can see the 4 digits. And where it's stained very dark, that's a marker for the areas where cell death is occurring. And so if you think about ducks that have webbed feet and chickens that do not, both of them go through a process of cell death during the development of the limb, but it's under control. And in the case of the duck, there's just a little bit in a few regions, and the spaces between the digits is mostly left unaffected. Whereas in the chick embryo, the programmed cell death extends much further, and it reaches down into these interdigital spaces and leads to the loss of the cells in there, so that the chick feet can have separated toes. And this is just an image of a chicken that had a block to BMP signaling in the interdigital space during the time when that programmed cell death was supposed to happen. And it's a perfectly fine chicken. There's nothing wrong with it, but it does have the webbing between the toes. And you can do some experiments to show whether it's the mesoderm or the ectoderm that promotes this. So the ectoderm of a chick embryo or a duck embryo in the limb bud formation. And if the chick ectoderm has chick mesoderm underneath it, you get the separated toes. But if you have duck mesoderm transplanted there, that is not going to go through the cell death, and so it creates this webbed foot. In the duck, the ectoderm combined with the chick mesoderm gives you a duck who doesn't swim very well. It's okay because it's being looked after, but it can't swim very well because it's missing the webbing, and of course, normal duck ectoderm and mesoderm gives you the normal feet. So if you transplant the chick mesoderm, the chick mesoderm below the duck ectoderm, then it promotes apoptosis and causes the separation of the digits, and vice versa, right? Okay, so this is a summary overall. So limb formation is triggered by those FGF signals in the mesoderm. And there are organizing centers for AP, proximal distal, and dorsal ventral signaling. Limb identity is specified by these TBX transcription factors. Remember, though, PITX1 for the hind limb is the real one that specifies that, and that these are related to Hox genes, but in some cases we just don't know exactly what the Hox gene targets are and exactly how they work. Anterior posterior patterning and digit identity requires both sonic hedgehog and BMP signals, and we went through lots of details about that. Dorsal ventral signaling is directed by the ectoderm and is much simpler than the others. Apoptosis is a normal part of sculpting tissue, such as the separation of digits, and in this case is signaled by the mesoderm. So these are all just a really basic summary of what we've been talking about in this section. So what I'd like to finish with is that there are organisms that are able to regenerate limbs. And so think about this in the context of being out in the wild. If a predator grabs you by the tail and your tail gets bitten off, that's not great. But if you can just regenerate another tail, then that's a very good thing. Same with a limb. A lot of poor aquatic organisms that might get grabbed by a fish, if a limb gets bitten off, it only takes a couple of months, I think, usually to be able to regenerate. These guys can also regenerate the lens of the eye and a number of other tissues. We're only going to be looking really at the regeneration process. And people have been studying this because the dream is that if we understand how this process works, um, after an accident, if someone has lost some fingers or lost a hand or something like that, what can we do to maybe promote regrowth of that? I mean, that's really the dream. And so that's where we're going to spend the final section of the class, which I really hope students will find interesting.